Hello again, and welcome back to another episode. As you can tell, we're moving on to cellular respiration. The second of our energy transformations, though this one's got a bit of a offshoot. We're going to go over two lessons, acknowledging that the last couple lessons have been nearly 40 minutes each. That's a long time for a YouTube clip, and a long time for you guys to listen to anything. So if this one pushes that barrier, we're going to be in a bit of trouble. But we've already been introduced to the general equation for cellular respiration. You may even know it already, but we're going to start by having a bit of an explore and have a look at the working parts of the cellular structures to do with cellular respiration, then move on to the equation and then how each step actually works. This is a very complicated diagram to introduce or start off with, so let's simplify it a little bit. So here's the mitochondria. And believe it or not, woohoo! I actually got it pretty close to a good diagram with my initial organelle drawing. I don't need to bring in the one from the internet. In fact, I can label this one to show each of the spaces. So the mitochondria does in fact have some parts, and in that first organelles video that I did, we started to talk about them. So first of all, it has this outer membrane, which is where is kind of the protective membrane. This is the one that contains everything inside of it, and we also have an inner membrane as well, much like the chloroplast. In fact, these guys are said to be pretty closely related to in their structures and also their origins. Both of these structures even have their own plasmid type DNA on the inside of them. And the theory is that these guys have bacterial origins or prokaryotic origins, and that they assimilated into a eukaryotic cell and became the energy and food providers for those cells. They have their own ribosomes and everything as well, so that's something that that's worth looking into, and we'll discuss that in a later episode as well. So between them as well, we also have this intermembrane space. This plays another role in cellular respiration as well. Much like it did in photosynthesis, where there was some of the hydrogen ions existing in it. All right, so these folds that you can see between in the inner membrane itself, these things are called cristae. It's a very fancy word, and this is where a lot of the action happens. In a lot of actual drawings and proper drawings, these folds are much closer together for mitochondria. They're very they're very close. They increase the surface area, so there's a lot of reactions that can happen between them and across them and things like that. So that's an important thing to note. And then finally, to talk about, we have the middle section here, and this is our mitochondrial matrix. This is another zone of happenings. This is the inner space, and a lot of things will be happening across these cristae in between across the inner membrane of the mitochondria, much like it happened with the chloroplast for photosynthesis. Okay, moving on. If you need a good graphic organizer, if you're looking for something to take notes on, for year 11s I generally use something like this, and then they can add extra notes to it. Again, even the year 12s can use this. This is one of those topics that's going to span across year 11 and year 12, so it's probably not a bad idea to have that information. I'm also going to go into a little bit more detail than the year 11s need, and probably just shy of what the year 12s really need. So use this as a bit of a stepping stone and start talking about all the bits and pieces from here. And again, if I get anything wrong, please, leave a comment in the comments section. We'll do the YouTuber thing at the end, subscribe to the channel. Haha. Anywho. All right, the equation. We have the start here, and you probably, oh geez, my animations have kind of cut off at the bottom there. But the black dots are representing carbons, the grayed out ones are your hydrogens, and the red dots are your oxygens. At the start of the equation, it's very simple. We start off with 6 oxygen molecules and a glucose, that's C6H12O6. This might help as well for those people who are thinking to themselves, well, I'm still not quite sure what you meant by having to balance the equation. Oxygen comes in and there's carbon dioxide and carbons and hydrogens. let's see what happens when the reaction actually occurs. And in chemistry science, we use an arrow to indicate a reaction, not an equal sign. There we go. I wasn't sure if that was going to work. Woohoo, animations. You can see here that everything is accounted for. All of those molecules moved over and there was nothing left behind. So at the end of our reaction, when we go through cellular respiration, we should end up with 6 carbon dioxide molecules and 6 water molecules as byproducts. Both of these are byproducts. Neither of them are used in at least in our bodies, in our cells for further reactions. In a plant will go ahead and use these. They'll continuously cycle in them. They're pretty self-sufficient. But, we don't use these products. What we're after, really, the really big ticket item is ATP. Cellular respiration is our energy producing reaction, and we're hoping to get that 36 to 38 ATP at the end of what we call aerobic cellular respiration. In a future lesson, we're going to go through anaerobic versus aerobic, so stay tuned. So let's talk about the steps of cellular respiration. We're starting off with step 1, glycolysis. Glycolysis means glyco-cutting. Lysis, remember that lysis word means to cut something, and so glyco being your carbohydrates, or in this case glucose, we're cutting up our glucose. This first step doesn't happen inside the mitochondria. It happens out in the cytoplasm of the cell. And it's around about 10 or so reactions that go from us having this C6H12O6 molecule to having 2 molecules of pyruvate. Now, I've added a little bit extra here. So pyruvate is 3 carbon molecules, 3 oxygen molecules, and 4 hydrogen molecules. For the life of me, through my research, I couldn't find out what happened to the other 4 molecules. So I'm just assuming that either they end up floating around as little H pluses out in the universe, or they end up getting picked up by something like an NADP plus in this reaction. This second half here, I'm not sure. Again, if you know, please let me know. Find it out and put in the comments. I'd love to be able to learn more as much as you guys do. Okay, so we have our 2 pyruvate molecules, and this is what we needed. We wanted to cut up our glucose into something smaller that we can work with. And now we need to check for a couple things. First of all, it's important to note that we had to use energy to get energy. So we started off with our glucose molecules getting cut up, and we used 2 molecules of ATP. So energy was used in this reaction. In the process later on, where pyruvate gets converted to another molecule, we gain a bit of ATP as well. In the process of making, in those 10 steps to make pyruvate, we gain some ATP. So we make 4 ATP. We get a net gain of 2 ATP. So a lot of the time, glycolysis will have outputs 2 ATP, when really we have 2 that come in and 4 that come out. It's just that we have a net gain of those 2 ATP. We also get 2 molecules of NADPH. Now, you're going to see that there's going to be a lot of these hydrogen carriers, these electron carriers. They're important for what we're going to need to do to these guys. All right. So now that we have our 2 pyruvate molecules, it's time to check for some oxygen. If oxygen is absent, think about it. You're doing some exercise. You're going for a really long run, and then you decide, hey, I'm going to sprint this hill. And so you take off down the line and you absolutely gun it, and you don't breathe in as much air. Your body is going to still try to produce energy for your muscle cells to use. The problem is that it's starting to use up its oxygen reserves, and so you are going to have an absence of oxygen. And this is where you end up having the pyruvate stay in the cytoplasm, and it gets converted to another type of molecule. Another lesson coming, we'll discuss that one. Today, we're going to be focusing on when oxygen is present. And so this time we're going to say that oxygen is present. You're going for that run and you're breathing in nice and deep. You've gone for a walk, these days, hopefully without a mask on, and you've been, no, that's a lie. Masks don't prevent the oxygen coming in. Anyway, so you've gone for a run and you're breathing in nice and deep. You're just breathing normally and your body has oxygen to use. So we're going to send these pyruvate molecules into the mitochondria, which takes us to step 2. Step 2 is known as the Krebs cycle or the citric acid cycle. Now for this one to work, oh, whoopsie, I forgot my bit of paper. 2 seconds. Beauty of a home office. All right, so this is where we're going to have our Krebs cycle, and for this to work, our pyruvate comes into the equation, but it doesn't stay as pyruvate. Before it hits the Krebs cycle or the citric acid cycle, it gets converted. Those molecules that you saw fly down there, please ignore that. This is for some reason part of the way the animation worked. I couldn't stop it from flying down there the way that it did. So we actually end up with a molecule called acetyl-CoA. This is acetyl coenzyme A. And it's another carrier. Coenzyme A is a carrier. It's going to take a couple of these carbon molecules and oxygen and a few of these hydrogens, and it's going to put them into the Krebs cycle. As you can see, the Krebs cycle has a number of stages. It starts off with this 6 carbon molecule, and it chucks in a couple other carbons as it goes. This is the carbon molecule carrier. This one here is citrate. Yeah, this is probably where it gets definitely where it gets the citric acid cycle, because it goes through this process of breaking it down and reforming it, much like the Calvin cycle when it exists in the photosynthesis. Next, we should also mention where this step takes place. This step takes place inside the mitochondrial matrix. So we're inside the mitochondria, and we're actually in that inner matrix part of the mitochondria. That's where this is taking place. All right, so this cycle comes along. You can see here that the NADP plus, comes along, grabs a hydrogen. This NADP plus comes along, grabs a hydrogen. This one comes along, grabs a hydrogen. Now, yeah, I've missed a little part here. This isn't FADH. This is FADH2, because FADH is another carrier molecule which has the potential to carry 2 hydrogens, not 1. And so it does this for each of the pyruvates. So you end up with 6 NADPHs, 2 FADH2s, and 2 ATP molecules. Again, you've got 2 pyruvates, 2 acetyl-CoAs, and so this happens twice. Boom. We've got a really good net stock of all this stuff happening now. We've got a lot of NADPHs, we've got a couple of FADHs, and we've got a little bit of ATP that the cell can use. At the moment, we're probably, we're sitting on a net of about 4. It's nowhere near the 38 that we had said we were promised, and that's because all of these molecules of NADPH and FADH from the glycolysis and the Krebs cycle are now going to come into action. And here we are at the electron transport chain. We've seen something like this before, and this works almost identically to this. So this is where I'm going to bust out my pencil and we're going to do a little bit of drawing. There's 2 membrane layers here, and that's because this here that you can see, this space here, this is the intermembrane space that we were talking about before. And the intermembrane space is where we're going to have a lot of storage of a particular ion in a second. So this here is your outer membrane, and this here is your inner membrane of your mitochondria. So the inner and outer membranes now exist, and or are labeled, I should say, and there's the intermembrane space. I said it was going to be an important thing. Each one of these proteins that sit in this membrane have a role in the electron transport chain. I'm not going to go through their names or anything like that, but I am going to give them some labels. This one's going to be 1 or I. We'll call this one Q. We'll call this one II for 2. And we'll call this one site, so C. And this one can be number 4. Sorry, Q and 2. This is Q and 2. My apologies. And this is number 3. All right. We'll get to this last one in a minute because I think you might actually know who we might be, but we'll get there. So we start off with an NADPH. Comes along, gives up the hydrogen, or detaches from the hydrogen, becomes NADP+. And an excited electron goes cruising through from here, and that excited electron forces a H+ into the intermembrane space. All right, I think if you remember how photosynthesis work, you may be starting to realize where this one's going. So there's our H+ out in the side in the intermembrane space. The excited electron has traveled along and then attached to the Q protein, or the number 2 protein. Here, this one here, this protein here is where FADH2 comes along. It gives up its excited electron as well. It becomes FAD. I'm really bad at spacing out where each one's meant to be going. And that excited electron, those 2 excited electrons go cruising up to site C past number 3. Now there's no NADPH getting separated here because once it goes past number 3, it grabs those excited, the excited electrons grab or force those hydrogens from FADH2 out of the matrix into the fluid. All right, so we're getting a collection going on here. There's a fair few that are starting to come into play here. And this excited electron, doesn't lose its excitement, but it does pass through this fourth protein. This fourth protein does 2 things. Firstly, it pushes out another H+. Once that H+ gets pushed out, there's an electron floating around in here that needs somewhere to go. And this is where our water byproduct comes in, because we end up taking 2 H+, and 1 oxygen, so 2 H+ and an oxygen, they grab that excited electron and become a H2O. So the H2O, the water molecule, is just a storage facility or a storage place, a carrier of electrons. Both before and after, if you think about it, the water molecule was split in a photosynthesis, and that is what gave us our electron to become excited, and then, the H+'s all got pushed out again. Alright, so as it turns out, we now have very few H+'s on the inside of this membrane, but lots in the intermembrane space. After a while, or after the 2 pyruvates come along and do this, go through this process, and we end up with double the amount of H+'s going on in here, we're going to end up with a fair few, or a large concentration gradient, which leads us to this last molecule here. So H+ comes through this molecule here, or this protein. By the way, that is one source of where the H+'s come from to make water. I should have made that clear before. So they come through this last one here, and as some of you may have guessed, this is ATP synthase. Now think about it, there are a lot of hydrogen molecules out here. And so for one glucose molecule, multiply this by, we have 6 of these, you're going to have 2 of these. There's going to be a fair few opportunities for hydrogen molecules to be out in the ether. We're going to end up with it coming through and spinning, getting some ADP plus PI, and creating ATP. All right. After this process, and I'll leave that up here for a second because, again, as my software dictates, if I want to show another slide, and I do have some more to show here, I have to get rid of my drawings. Annoying. As this process shows, we can end up with quite a net amount. So we're going to end up with 32 to 34 molecules of ATP after this process. This is where the bulk of our energy comes from. There's a bit of a discrepancy in how much we're going to get, and a lot of biologists and scientists try to explain this, but the basics behind it is that because of the usage of energy beforehand and the fluctuations with how efficient the Krebs cycle is and the cycling through, we're going to end up with a different number each time. So it's somewhere in the realm of 32 to 34 being made in this process, but ultimately it doesn't matter. You can say 36 and it'll be correct, 36 as the total number in yours, and it'll be correct. But, just in case you needed to know, why is there that discrepancy between them? That's why. Okay, as promised, I needed to keep it to be a shorter lesson. It is a shorter lesson. We discussed the mitochondria. I gave you a graphic organizer that you could fill in. Feel free to draw that and fill it in. I gave you the chemical equation, or hopefully the graphic should kind of help explain balancing of equations as well. That's something a lot of high school students kind of struggle with. I gave you glycolysis, Krebs citric acid cycle, and the electron transport chain. And I'm just tipped in at the 20-minute marks. The 22.5-minute mark. Oops. Anyway, enjoy, and see you in the next one.